Today we're going to be going over AP Biology Unit 3.1, which is going to be all about enzymes. So in this video, I'm going to be talking about enzyme function, enzyme structure, and something that we called an enzyme inhibitor. So what an enzyme does, enzymes are what we call a catalyst for reactions. And what a catalyst does is that it lowers the activation energy needed to start a reaction. So activation energy is the level that a reaction needs to reach until it actually gets to start. So all reactions need to reach this activation energy in order to start. And what an enzyme does is that it's a catalyst, so it helps lower that activation energy. So you can tell from this image right here, we have, this is the red line is without the enzyme, and the blue line is with the enzyme. So what happens is, this is the activation energy. The reaction has to reach this tippity point before it can actually carry out the reaction. So what happens is that when you have an enzyme, the activation energy is shortened significantly. If you don't have an enzyme, it's very, very long. So in our actual bodies, it's very rare for a reaction to be able to start without any sort of help because this activation energy just takes way too long. And oftentimes reactions are useless if you have to wait that long in order to start them. Okay, so an enzyme, how it functions is through what we call an enzyme substrate complex. So a substrate is really just a fancy name for the reactant that the enzyme will act on and speed up. So when an enzyme and its substrate form together and they bind, we call that an enzyme substrate complex. And the enzyme will act on the substrate and produce a product, much like a regular reaction. So this is a diagram that it looks like the E and the orange is considered the enzyme, and the S or the blue is considered the substrate. So this part where the substrate is binding to the enzyme is what we call an active site. We call this an induced fit or lock and key structure. This is because this is an almost perfect fit. The enzyme and the substrate will bind together almost perfectly, and it'll be very unique to that enzyme and that substrate. After it produces it, you can see here it turns into product 1 and product 2. All right, so we have some outside factors that can affect the enzyme. And this is mainly because enzymes are considered proteins. And if you remember from my macromolecule video, proteins can get denatured. So proteins get denatured when temperatures are too high or when a pH changes out of its normal zone. So an enzyme, much like other proteins, can get denatured. What happens when an enzyme gets denatured is once it gets denatured, it will slowly lose its function. But until it's actually denatured, an enzyme will still function, just much slower. So when a temperature increases, enzyme activity will actually increase with it. But once the temperature reaches a certain level, the enzyme will stop completely. That's the same thing with pH. But pH, pH on the pH scale, if it changes just slightly, it can have a really big detrimental effect. So it's hard to tell for pH because every enzyme is different. But in regards to temperature, enzymes are pretty common in that once the temperature increases, it will increase in its productivity. But then once it increases to a certain point, it will completely stop. All right, so we have what we call inhibitors. So inhibitors are what we consider something that inhibits the function of an enzyme. So we have three main kinds of inhibitors. We have competitive inhibitors. These are the inhibitors that bind directly to the active site. So it binds directly to the active site and prevents the substrate from binding. And if the substrate can't bind, the reaction can't occur. If we have a non-competitive inhibitor, that's where the inhibitor will bind to another site on the enzyme. So not the active site, but once they bind to the other site, it changes the shape of the active site and the substrate will no longer be able to bind. I have a diagram. I know the non-competitive inhibitor is kind of hard to view in your mind. I have a diagram coming up that will explain it much better. And then we have the last one. It's what we call a feedback inhibitor. And this is where the final product of the reaction becomes the inhibitor of the same enzyme. So what I mean by that is, say lactate, for example. Let's say your body wanted to make 10 lactate. What will happen is they will make those 10 lactate, and then once the 10th one comes out, the body will be like, okay, we don't need any more lactate. What will happen is now the lactate will become an inhibitor. So the lactate will inhibit its own enzyme, and that will stop any further production of the lactate. So here's a diagram of the three of them in action. So we have a non-competitive inhibition. This is the active site, and the inhibitor will bind to another site. It will bind to not the active site, but just any other site on the enzyme. And what will happen is, you can see it changes the shape of the active site. It's no longer this deep V, it's more like a curve. And now the substrate can't bind to it like it originally would. So that's what we call a non-competitive inhibitor. It's where it will bind to another place, but it'll change the shape of the active site. And then we have what we call a competitive inhibitor, and that's really just where an inhibitor of the same shape as the substrate will bind here and prevent the substrate from binding. The end product inhibitor, you can see this is the substrate. The substrate comes out as a product, and then the product becomes an inhibitor. So these are the three main types of inhibitors. These are the only three that we really need to know in biology, AP biology. Yeah, so that's the end of this video. I hope you come back for my next video. We're going to be talking about Unit 3.2, AP Biology.